What's up, everybody? How are you doing? Welcome to Joon's channel by Jonathan Petrozzini. I'm your host today, Jonathan Petrozzini. Let's get right into it. So the topic we'll be discussing today is protein absorption. Scratch that, it's actually adsorption. Adsorption is defined basically as the accumulation and adhesion of things to a surface, but without surface penetration occurring. Now you may be asking yourself, why does this even matter? Protein absorption is the first process that occurs after implantation of a biomaterial in the human body. This process changes the properties of the surface and can induce structural alterations on the absorbed proteins. Cell biomaterial interactions are mediated by the type and conformation of absorbed proteins that can interact with specific integrins expressed by the cells. So in short, it's very important to biomaterials. So there's three main components that go into protein absorption. You've got your protein, your surface, and your environment. We will explore these aspects one by one to see how each of them affects protein absorption. So let's begin with the protein. There's two main factors that affect protein absorption. There's the surface affinity of the protein and the concentration of the protein. The higher the concentration of the protein, the more likely it will be in contact with the surface and therefore will attach to the surface more. And the surface affinity is basically how energetically favorable it is for a protein to be absorbed to a surface. Now the surface affinity is also affected by many components such as the size, so the larger the protein the more areas of contact it has with the surface, and also by the charge, so depending on the surface the protein may be attracted or repelled, and more hydrophobic proteins tend to stick to surfaces in greater amounts. Additionally, the structural stability of the protein affects how much it absorbs to a surface, because the less stable it is, the more it will unfold and the more contact points it will have with the surface. There's even more factors that affect the structural stability, such as the side chains. So if you have a lot of different hydrophobic and hydrophilic side chains, it might coil up and not really unfold, and the unfolding rate will determine how quickly a protein is absorbed. Now moving on to our surface, there are many surface properties that affect interactions with proteins. First of all, there's surface potential, which will influence the distribution of ions in the solution and interact with the proteins. Next we have topography, because greater texture exposes more surface area for interaction with proteins. Next we have the composition of the surface, because the chemical makeup of the surface will ultimately determine the types of intermolecular forces that govern the interactions with the proteins. And we also have the hydrophobicity of the surface, because hydrophobic surfaces tend to bind to more protein. Next we have the heterogeneity of the surface, because non-uniformity of surface interactions results in domains that can interact differently with proteins. And the final component we have to look at is the environment that the protein and surface are in. So the pH of the environment will dictate how close the protein is to its isoelectric point, and the closer the protein is to its isoelectric point, the more readily it will be absorbed. Another factor in the environment that will greatly influence protein's absorption to a surface is temperature, as it is closely tied to the diffusion, thermal convection, and flow transport of a protein. As you can see, there are many factors within the environment, the protein, and the surface itself that determine whether a protein will absorb to a surface. This process is so multifaceted, you may not be able to predict whether a protein will absorb to a surface until you actually try it out yourself. Now that we understand why protein absorption may or may not happen, let's see how it changes over time. So now we are going to look at the total amount of absorbed protein to a surface over time. There is an initial high absorption rate correlating to the diffusion-controlled mechanisms. Next, there is an overshoot or a hump in the adsorption curve that can be explained in a variety of ways. For example, it may be the rearrangement of the proteins on the surface, or it may be that the proteins that are less strongly bound may desorb, which is known as a short period of reversible binding. Then after some time, a plateau value is reached. So if this is the protein absorption curve for one protein, you may be thinking what multiple proteins may look like. Well, because proteins may have different surface affinities and different concentrations, they will have some sort of exchanging on the surface, which is known as the Vroman Effect. So an analogy that can be made to understand this effect is people looking for a spot to sit in a park, where the people are the proteins and the spots in the park are the surfaces. So say the park opens early, and there's a lot of little children that go, and they have high energy and there's a lot of them. They are going to take up most of the spots initially. These children represent the proteins in high concentration with relatively low surface affinity. But then the adults start to come into the park and they demand their spots from their children. They ultimately displace the spots from the children. The adults in this metaphor represent higher surface affinity proteins. Then the sumo wrestlers come and they demand their spots from the adults. They ultimately end up displacing the adults and the children that remained. In the end, there are only authoritative sumo wrestlers residing in this park, and they are happy. These sumo wrestlers represent the highest affinity protein. So as a recap, proteins with higher concentrations will rapidly attach and then be replaced over time by proteins with greater surface affinity. And here's a little summary slide for those of you studying for the prelim post-pandemic. Thank you for tuning in to Joon's channel, and I'll see you next time.